This is an image of what's soon to come at University of Washington in Seattle. So normally right about mid-March to beginning of April, our cherry blossoms bloom. And our lab is just to the right, like basically adjacent to this fountain. So if you want to come check us out, come visit, say hi in Seattle. We'd love to see you. And I always like to, advertise. People don't always think of this Washington for the cherry blossoms, often Washington, D.C., but we both have them and they're quite a sight to behold. Also with that, I'll start too with another image of this same view. This is actually from a week ago, and so it's not quite the cherry blossom season yet. But I also wanted to acknowledge the land that we occupy and the indigenous peoples whose land was who was here first. So this is a land acknowledgement statement. And then also I feel compelled to make a statement just on the elephant in the room of current events and some attacks on science and free speech. And this is totally me only, not any of my employers, not my group. But this is just a quote from the poem that a lot of people have heard before by Dylan Thomas, just to not go gently into the night. And so I think as we face some challenges, I hope as scientists we can continue to fight for our science and fight for the scientists that we support. So that's all I'll say, but I know it's something on everyone's mind. So I'd be happy to connect with people afterwards if they want to talk more. But okay, with that, let's dive into the group and then the science that we're doing. And so been really fortunate to build a really strong group so far, and we have people whose work will be highlighted throughout this talk. And so I wanted to make sure to show faces first, and that includes work from Dr. Emma J. Sutherland, Katie Cothlow, Jacob Russell, but really almost everyone on this slide will have touched data that you're seeing today. So just wanted to make sure to acknowledge that. So I'm excited to share their work with you as we work together in the Riley Research Group. But okay, so I didn't put too much of a background on glycosylation in this talk because I figured most people are tuning into a Glygen webinar have some perspective on why it might be important. But I always love this image. It's from a Matt Pazik review that just shows how crowded the cell surface can get. And there's not any resolution on what these molecules are, but we know these are large glycoproteins, and they kind of represent how complex and heterogeneous the cell surface is. And we call that the glycocalyx, of course. And this glycocalyx, we've seen a lot of images where you have these, Lucky Charm figures. I assume most people on the call have probably seen something like this before, but that these, circles and shapes and colors, they all represent chemical structures. And those chemical structures directly affect the function of the proteins they modify. And it's this combinatorial glycan plus protein epitope that drives a lot of this function that we care about that's at the cell surface. And so here you can see this is images of staining for the glycocalyx, and it's such a dramatic feature of cell surfaces that clearly we want to understand how this is going to affect interactions of a cell with its environment. And so we want to be able to dial in and really be able to measure what glycoforms of these proteins exist and how we can understand how they contribute to interesting functions that we care about. So with that, my group thinks a lot about how to study this really complex cell surface glycoproteome. And there's a lot of crosstalk between the biology and the cell surface questions we're interested in. And also, how do you even begin to measure this really heterogeneous and complex glycocalyx? And so we like to think that we live somewhere in this interface, the crosstalk between them. But I also think most people that have done method development, especially in the glycoproteomics world, but I think just broadly in the mass spec world, can appreciate that it's not quite this linear of a track. It's more something like this, where each part of a workflow, like how you prepare and collect your samples, how you get them ready for mass spectrometry, the methods that you use, the data that you use to analyze it, they're all informing each other. And you have to make smart decisions about which tool you would use in any given case. And importantly, we in the group, at least, we like to center our biological questions as the driver for some of the methods that we want to build. And so hopefully I can communicate some of that through the time we have today. And hopefully we can have conversations where you could also benefit from looking at your glycoproteomics data with some tools that we are trying to make available. All right, so with that, I'll just, broad overview of things we think about in my group. We think about how to enrich and capture the complex glycoproteome. We think of that workflow we just showed in the previous slide, all the methods that go into characterizing that. And then ultimately, we're really interested in using these methods to study the structure-function relationships of glycoproteins and how they interact with receptors at the cell surface. And also, our lab is particularly interested in the regulation of glycosylation in metastasis, and not only the proximal regulation of cell surfaces, but also the distal regulation of biomolecules that get secreted, which we often know are also glycoproteins. So this is a big scale, 10,000-foot view of what we're working on in the group. We're going to spend most of our time in this space today, but I look forward as the group continues to grow and to mature in our projects to spend more time on this half of the slide in the coming years. But right now, we mainly methods focused, as the title of the talk probably gave away. So I'll start first with some methods of how to look at glycopeptide tandem mass spectra. And this is really the heart of the glycoproteomics world, how we get identifications, how we can try and discern glycan composition, maybe even glycan structure. And so if you've looked at a lot of tandem mass spectra of glycopeptides, this is immediately recognizable. Even if I didn't have these labels, you'd be like, that looks like glycopeptide, mainly because we have some dominant features, things like oxonium ions, and these tend to be glycan-specific, sugar-specific fragments that let us know a lot about what sugar could be present on a given species. We also have peptide fragments, and these are, the B and Y or C and Z ions that allow us to sequence the peptide backbone. And then also we get these large Y-type ions that can give us some information about the glycan composition and sometimes also even structure there. So this is kind of a home base of what we're thinking about today and some of the tools that we're trying to develop. And we're going to really focus mostly in the first part of this talk on oxonium ions. And again, these are the sugar-specific fragments. And we're not really the only people thinking about this, right? This is just not even an exhaustive list. People have been thinking about oxonium ions and how to use them intelligently for a long time. And so we are just adding another tool into the mix and are excited to kind of join the interest that the community has in this space. So if you were to look at an actual tandem mass spectrum, kind of what type of oxonium ion information do we get? And so this is taken from a spectrum from a publication. And so you can see here that we have some of these Z ions that are listed. Those are peptide fragments, but everything that's annotated here, you can get a lot of information about what sugars might have been present on this glycopeptide without knowing really anything more. Like we can see some features that look, oh, there's a fucosylated lacnac. That's cool. We also have, some, uh-oh. Someone okay? Okay. We also have some acylated glycan fragments, so it gives us a lot of information about what our glycan might be. There's a whole host of what these ions could be. And again, this is far from an exhaustive list, but it's one that a lot of people might start with when you think about hexnac ions or hexose ions. And again, this could be glycnac or galnac. And there's so much, the structure or the stereoisomers of these sugars that sometimes we can't tell or sometimes we can. But these ions are present in a lot of tandem and mass spectra. And so we want to be able to investigate them and use them to understand our data better. And things that you can answer, questions at least you can ask when you have these oxonium ions present, it can tell you, first of all, do I even have glycopeptides in my sample? You can start asking, like, do my glycan types differ? How well did an enzyme, like a glycosidase or a glycosyltransferase, work? Because I can look for ions that might show that it took away or added something that I was expecting. Potentially, you could ask what structural features exist on the glycans of my glycopeptide. And also, thinking about workflows and method development, how did some upstream workflow step affect the ability to detect glycopeptides? And, you can imagine there's a lot that we can understand just by kind of digging into some of these ions. So if you were to go look at raw data, so this is raw files from a thermo instrument. And so if you were to open up those files, you can start then looking at a total ion chromatogram as this top black trace. But then each of these color traces has a different ion mass to charge. And so these correspond to some different glycan masses. And so what becomes pretty apparent is to say, well, we definitely have glycan-specific fragments. But the challenge would be like, how often do they co-occur? How often do we have 204 and 274 in the same spectrum? Or what fraction of the total MS-MS scans are there sugar ions? And so it's easy to see these, but really hard to kind of extract and do more interesting analyses with them. And I'll mention too that, we know that search algorithms make use of this information. So here's just screenshots of a few, by far not an exhaustive list of the tools that are available, but you can see here where you can enter in some oxonium ion information or check boxes that look for oxonium ions. So search algorithms are using this, but often it's tied to an identification. And that's not a bad thing. They should be used when we try to identify glycopeptides, but also we'd like to be able to access them without the need to couple that to an identification of a scan. And one reason for this would be, let's imagine you took this many MSMS scans in just a given LCMS run. And so here, the total number of MSMS scans is gray. And so you have some fraction that you identified that were glycopeptides. And you could have used any of these search algorithms to identify that purple bar. Now, a question would be, what are the non-identified MSMS scans? If you look at something like this, and you could say, well, these are only a small proportion of the total had oxonium ions in the first place, and we identified a pretty large chunk of those. That's really helpful information to know, like, well, I searched the data correctly, but maybe I just need to improve my workflow to improve the enrichment and maximize the number of glycopeptides that are entering my mass spec relative to the non-modified species. And this is a very different situation than if you were to say, well, look at all of these MSMS scans that have sugar ions, and I'm identifying some really small fraction of them. Maybe I'm not searching this correctly, or maybe the way that I've approached this, I'm leaving a lot on the table that I need to reevaluate. And so this is kind of the information that we don't have a ton of access to at the moment that we'd like to improve. And I'll also say it's helpful to think about this in the context of overlapping or really lack thereof overlapping identifications from these search algorithms. It's a great study in 2021 and more studies that are ongoing now to kind of compare all of these different glycopeptide search algorithms. And this is not a figure for you to really dive into the details, but just to kind of comment that the overlap was really, really low. And so even if one search algorithm gives you the purple bar, maybe another search algorithm could give you more of the teal bar, and that can help, again, you know that that's there in the first place because you were able to look for these oxonium ions. So again, the challenge of like looking at this is you would not be able to comment on the teal bar just by looking at this data. So instead, our goal was we really wanted a way to extract multiple oxonium ions simultaneously and to get that oxonium ion content from raw data. So we didn't want to couple it to a search. We'd like to do it independently of how we identify glycopeptides. And more than just knowing the presence of them, we'd love to be able to evaluate their abundance and signal to noise, maybe the peak depth, because if they're not a super prevalent ion, maybe it's not as useful of information for when we try to identify glycopeptides. And so the goals in this, again, quickly evaluate spectral quality and inform us about glycan content. A really big reason is we do a lot of method development in the lab. And again, it's really helpful to check just a sanity check of after you collect data before you identify anything and say, did we lose or gain a lot of glycopeptide signal based on the oxonium ions that might be present? And finally, also supporting spectral assignment. So if we have challenges with how the overlap or the identification rate of our identification software, it'd be nice to have ions to go look for and use as a cross-reference to make sure that we can understand how those search engines are working. So with all of that, we generated this tool called Glycounter. And this is, again, I don't want to pitch this as anything sophisticated. It's an incredibly straightforward and simple piece of software. We found it to be highly useful for a lot of applications that we're thinking about in the lab. And so how you can use Glycounter, you upload raw files or MZML files. So hopefully, cross-compatible across a lot of different vendors and platforms. And you can check any ions that you are thinking you might want to look for relative to certain sugars. You can also even upload custom ions that you might want to look for as well. And you have a lot of parameters that you can set to determine, how you extract this. And so it gives you just a lot of knobs to turn that gives you, access to information. And we have defaults and things that we set, but you can always tune these and use this to explore your data. And so the way I like to phrase this, all that changed when I advanced that slide was the title here, like, what are your ions telling you? That's basically what I envision Glycounter to be. Just listen to your ions. And so I'll give quick vignettes and we'll move on to a few other things that we're thinking about that Glycounter helped enable. But to show you some examples of how Glycounter can help with glycoenzyme evaluation, look at modified glycans and modifications you might not expect to see, how you can really rapidly glycotype tissues, and even look at glycopeptide fragmentation and other method development angles. So I'll just give you a few quick examples of how this looks and how it can be useful. So the first is the glycoenzyme idea, and a class of enzymes I worked on during my postdoc and I still use often, and is really a cool and emerging class of enzymes are these O-glycoproteases. And what's cool about these is that they require a peptide plus glycan motif. So they cleave only when certain serine or threonine residues are modified in the right way. And the challenge is to understand what are the motifs. And so we have to often do this empirically. It's hard to predict. And so you have to do a lot of tandem MS experiments to define where these enzymes might cut. And this is, it's a growing list. So some of these enzymes are specific for mucins, some are not, but will cut at any O-glycosylation site. And so we really want tools to be able to explore this data. And also, again, because we have to collect so much LC-MS data, it'd be really nice to, be able to expedite how we can explore what information these enzymes can give us. So one example of this would be something we worked on still in Carolyn's lab. And so this is data that's publicly available that we just pulled as an example. But we basically have these four recombinantly expressed glycoprotein standards, so to speak. Now, standards, of course, a loose term, but things you can recombinantly express and then digest with any of these O-glycoproteases. And things that we really want to explore would be like, well, does it matter if a sialic acid is there or there or there? And really explore some of the glycan features that might drive how these enzymes perform. And so often we might do a sialidase treatment and compare how the enzyme cleaves with and without sialic acids present. And so a really important question that you should always ask yourself when you're doing PNGSF or sialidase or like any glycosidase or glycosyl transferase is, how do I know that it worked? And so an easy way would be to use Glycounter and just look for ions that show you sialic acids. And so here we see these are sialic acid specific ions for new AC and new GC. And when we treat with our sialidase, they all but disappear. And this is percentage of MS spectra where they were. And so this gives us a pretty confident idea that sialidase did remove sialic acids, and any cleavage differences we see from these glycoproteases is driven by the treatment of the sialidase. Other things that are interesting just to point out really quick is also we were surprised to see new GC because all of these are human proteins, and often we don't think of human proteins carrying new GC. And so, of course, the smart thing would have been to check this beforehand, but as we're moving quickly for some experiments, we went back and looked and saw like, okay, well, MUC16, the orange bar and the green bar PSGL1, those were expressed in CHO cell lines where we don't often have new GC being generated. Whereas CD43 and GP1B alpha both were expressed in NSO, which is a mouse cell line. And so, of course, now we see new GC in the mouse cell lines, which gives us some information and useful metric, first to just rationalize why we see those things, but also when we originally searched this data and tried to analyze it, we didn't include new GC just because, we hadn't thought to stop and think about, what types of glycans should we make sure to include in our glycopeptide search. And so you can see if you use this as a quick check to say, oh, these ions are present, you can make sure you search with the right types of glycans, and for your glycopeptide identifications, you can gain, in this case, 30% of IDs. So is it, again, just a useful quick spot check before you go down any further data analysis. Other things too, I want to quickly talk about some glycan modifications and glycotyping of tissues. So first, I'll show you this really cool paper that came out last year, so 2024. It looked at O-acetylation of sialic acids. So it's not just that something is sialated, but that those sialic acids have that extra acetyl modification that's on them. And so this study was cool to look at serum glycoproteins between human, rat, and mouse. And so they saw interesting patterns that we, basically took from their supplemental and just replotted right here. So this should be similar information, just in a slightly different order. But you can see that human had a lot of sialated and some non-sialated glycans. Mouse had, a lot of sialated, some acetyl sialic acid, and some non-sialated glycopeptides, and rat had a huge proportion of that acetylated sialic acid. And so this was all coming from identifications after a search. But a question could be like, could I understand this data without having to identify anything? Could I just understand what my patterns might look like for these sialic acid modifications before I search it so I know how to search it properly? And so first you can look at the percentage of MS spectra with different NUAC ions. So here we're looking at new AC and then new AC with acetylation. You can see in human, there's almost no acetylation of new AC, and in mouse, surprisingly, almost no acetylation of new AC. So it's interesting that we have that yellow bar there. But then in the rat, you can see that there's a high proportion of spectra that have the acetylated sialic acid as well. So it kind of explains that yellow bar. And then if we come in here and add in new GC, it's immediately apparent that all of the acetylation in mice and mouse serum glycoproteins is coming from new GC acetylation instead of new AC. And similarly, in rats, almost none of the acetylation signal is coming from new GC, almost all from new AC. And so again, this graph told us a lot, this bar graph, without having to identify or search anything. And so it kind of gives us perspective on things, the glycopeptides we might expect to identify in these types of samples, and again, what glycans to use when searching them. Another idea is glycotyping tissues and evaluating search algorithms. And so I want to point out how incredibly valuable this pGlyco 2.0 data set has been. So this came out in 2017, I want to say, and it's just used as a great benchmark for a lot of search algorithms. And so one thing we decided to look at is if we, they have different tissues that they evaluated in this, and we'll get to some of these percentages here in a second. But we just looked at the heart and the brain as an example. So tissue types here. And we were curious if we looked at with Glycounter, how many different spectra would we expect to be glycosylated based on the presence of a collection of oxonium ions? So you can say here that there's two different settings that were used in this, but you can see there might be like 25,000 to 35,000 spectra that you expect to be identified in the heart data set. And then we can look at the different search algorithms as people have developed and tried to improve them. How many of those spectra do we actually identify as glycopeptides, and what are we leaving on the table? So you can see here in the heart tissue, there's still spectra that could be potentially glycopeptides that remain unidentified. While in the brain tissue, we kind of see, like, depending on how relaxed a search algorithm or how well that it's identifying glycopeptides, you might see that they're identifying more glycopeptides than even would be expected based on the presence of oxonium ions. Now, I want to make sure to say this likely glyco is a human decision. Like, we are choosing to say it needs this many combination of glycosylation or oxonium ions to tell us that it is likely glyco. So it's not that this is necessarily wrong, but you can get kind of a snapshot of what proportion of spectra would I expect to identify and what am I actually identifying. Similarly, in the same data set, they saw in kidney tissue that there's a ton of fucose. And so we were interested to look and what oxonium ions could we look for that could explain the amount of fucose that's there. And so you can see that this dotted line is the number of fucose identifications that were explained by pGlyco. And you can see here the bars show in different replicates what fucose-specific oxonium ions that we could see in that data set. And so, what could be useful for this. I'll mention too, I haven't yet shown, but the Glycounter can also identify large Y-type ion glycans. And so here, you actually have to have done the identification, but you can load that into Glycounter and then look for peptide plus glycan fragments to start understanding what could be there too. So then again, this is the top one is being able to evaluate this without doing any searching. And the bottom graph here is after you've done a search, could you go look for ions that you expect that could explain some big trends you see in your data. And then finally, for this data set, also is interesting to look at, looking at the proportion of new AC and new GC that they see between different tissues from mice. We can recapitulate some of those patterns, again, only by looking at oxonium ions. So here's a new GC set of ions. You can see it's effectively empty in the brain, and that matches the identifications that they got. So again, this kind of helps you couple a sanity check of did my search algorithm identify the right thing? Well, it's explained by these fragments that I expected to see or not. So the last thing I'll highlight for Glycounter, and then we'll move on to one more set of stories about method development and maybe how Glycounter helped us advance things, would be to look at glycopeptide fragmentation. So we have two quick stories about this that I want to show you. And so one of them is a study I did while I was still a postdoc in Carolyn's lab. It's actually one of the first studies I did in the group because I was so curious. I really wanted the same dissociation method to be useful for N-O glycopeptides. And it turns out it's not always the case. And so we'll revisit this idea in the coming slides. But one thing we did is we set up a whole bunch of different collision energy settings with HCD or ET-HCD, which would be electron-based fragmentation with supplemental activation, and even stepped HCD method. So we set up a whole panel and we investigated this. And so here, we're looking at, well, if I wanted to generate the most signal from my glycopeptides, the most oxonium ion signal, you can see here we have percent tick. And so Glycounter, without searching anything, allowed us to just pull out all of the oxonium ions we see across all of these identifications. And you might say, well, these HCD energies, which are kind of these energies that are right here, maybe those are great for maximizing the amount of oxonium ions we see and maybe potentially the amount of glycopeptides we could identify. But as it turns out, it's always important to look for multiple of these ions. And so here you can see sialic acids. These are like the 272 and 274 and 292. You can see that you start really breaking those away as you go to higher collision energies. Well, maybe some of the stepped collision energies give you a bit more signal there. It's even more true for some of the larger oxonium ions. So here, this is the hexnac hex nuac. So this can be really useful for also identifying N and O glycopeptides. And you can see at the higher collision energies, what you might have chosen for total oxonium ion signal would totally destroy this ion. So if you wanted that as a useful metric, you would, could look at your data and understand how you're affecting what the results might be that you expect. And similarly for this other, trisaccharide ion that can be helpful discriminating in or different types of N glycopeptides from each other. So this is just one way, again, that no identifications are shown in this slide. It's all just extracting the ions that we saw across all of these different conditions and trying to make sense of what we saw there. Another really, really cool study that came out recently, so 2023, and it's been extended by Carly Reading's group further in the recent years, is to look at structural information that you can get as you do collision energy ramps, or like basically breakdown curves of glycopeptides that have different structures. So what they showed is that they could, basically take a glycan with this structure and across different collision energies, look for different glycan fragments and see a different proportion as you go to different collision energies. And so this gave them clues as to what the glycan structure is. And so we were like, well, they had specialized tools and were writing a in-house R script and for this, or maybe it was, Jupyter Notebook. I can't remember the details off the top of my head, but we were like, what if we could introduce Glycounter as a useful tool for this type of analysis too? And so here we just downloaded their raw data, and this is from Glycounter, just recapitulating the exact same data that we saw there. And so this is just showing, when you use Glycounter, it's easy to extract this information and make these plots yourself. And then similarly, they did another really cool study where they had between these different isomers, basically like what's the heat map of different glycan fragments that you might see across different collision energies. And so, the point is not to necessarily interpret this graph in detail, but just to show you that if you wanted access to that information, that with Glycounter, you can reproduce this with relatively high fidelity without even having to know what the peptide sequence was or the glycan in that case. So all you would need to know is be able to compare these two different structures that you might have been looking for in your sample. So, okay, that was a ton of kind of quick stories, but I just wanted to highlight when thinking about like the data informatics part of this wheelhouse, how it's informing basically all of these steps. It's helping us decide collision energies, helping us decide maybe how we prepare samples or even what samples we are looking at and what we might want to analyze. And so I want to tell another story of basically a direct information exchange between what the oxonium ions that we look for and how we built a new data acquisition scheme for N and O glycoproteomics in the same run. So with this, I kind of mentioned from that optimal dissociation methods paper from my postdoc, it became pretty clear that N-linked glycosylation, collision-based fragmentation is often sufficient. Now, I don't want to say better, but sufficient is a good word because you often can get the information you need, even if the spectrum isn't as high quality as you might be able to get with the electron-based fragmentation. But the collision-based fragmentation is super expedient, and so it's effectively instantaneous. So you can just collect more spectra, and this gives you a benefit for just, again, more shots on goal to identify glycopeptides. On the other hand, O-linked glycans and O-linked glycopeptides, the collision-based fragmentation really knocks off all the O-glycan signal often. Again, this is not like a hard and fast rule, but a general observation that we had from the data where we saw because there are multiple potential O-glycosites and a lot of peptides that are O-glycosylated, you really need electron-based fragmentation to retain the glycan and be able to localize where it is along the peptide backbone. Now, a challenge with this is that the electron-based fragmentation can take longer. And so, as we keep getting better instrumentation, this number is dropping, but tens to 100 milliseconds is pretty standard for what you would need for an electron-based fragmentation scan or to do that type of fragmentation. And so a question becomes, how do we do, oh, excuse me, I left it out, this, how do we only do electron-based fragmentation when we need to? And so one way that people have been working on this in the field is to look at oxonium ion fragments. And so we have the product-dependent triggering, where if you see these oxonium ions, you can start making decisions about collecting electron-based fragmentation spectrum, and that helps you get you information. But a question we had was like, could we understand if a MS spectrum we just collected, does it look more like an N-glycopeptide or more like an O-glycopeptide? And again, we are not the first people to look at this. We looked at that data that we had generated and said, we see this pattern where if it's an N-glycopeptide, the 138 to 144 peak tends to be 10 to 20-fold higher for the 138 peak. Whereas for O-linked glycopeptides, this tends to be closer to 1-to-1. And you can see the distribution here. And so our question became, like, could we, in real time, start looking at this pattern and decide as we're acquiring data if a peptide is N-linked or O-linked? And so I should mention, like, this graph, we extracted all of this data and made these ratios using Glycounter. And so this gave us direct access to kind of explore how this tool, how we could use this information to guide our method development. And so again, we're trying to use the oxonium ion signal we see to decide, is it an N-linked or is it an O-linked glycopeptide? And so to kind of juxtapose this to the traditional acquisition schemes, I just want to show you here, we have our MS1 and MS2. This is data-dependent acquisition. So if you have done this, this will be very familiar. If you haven't done this a lot, it might be a little confusing. But what this figure is trying to show is that you'll pick ions. So the first most abundant ion, the second most abundant, third most abundant, and you'll select those in order of abundance to then fragment with different collision energies or different types of tandem MS. And so here, this would be a stepped HCD only method, which is pretty typical for N-linked glycopeptides. And so we're showing a single HCD energy here, but you could do stepped, you could do a lot of different things. And you're able to get X number of tandem MS spectra per cycle before you take another MS1 and start picking other ions. When you do this combination that currently exists, it's kind of like what most people would do if they needed to do ETD, is they do a collision-based fragmentation and an electron-based fragmentation for each precursor. And you can check, like, I only want to do the electron-based fragmentation if it's a glycopeptide, but you still don't have the ability to say, is it an N-glycopeptide or an O-glycopeptide? And so what ends up happening is you do HCD and ETD for the same precursor every time. So you see this glycopeptide exists, so let's do HCD and then ETD. And this glycopeptide exists, HCD and ETD. And as you can tell, we get less, we hurt our sensitivity by doing this because we can dig less deep into the glycoproteome. And so a goal would be, how could we choose what N-glycopeptide or O-glycopeptide might be through something we call a real-time library search. And so this is embedded on the instrument computer. And so if you have a Orbitrap Eclipse or Orbitrap Ascend, this is available to you. You have this that you could try yourself. You basically did a really simple comparison of our experimental spectra to theoretical N and O glycopeptide spectra. We calculate a cosine score with this real-time library search, and then we send that to a filter. And the filter decides, oh, it's an N glycopeptide or it's an O glycopeptide. Choose what fragmentation to do. And right now we're at the 3 millisecond time scale. It's actually quite substantially lower than that now. So it's really a negligible amount of time to do this comparison to then send it back to the method to make a decision for fragmentation. So what this looks like, if we put this in perspective with the other two more traditional methods, is this idea of autonomous dissociation type selection, or ADS. And so here what we're doing is we take an HCD scan and we do a real-time library search to compare those oxonium ions. If it looks like an N-glycopeptide, we don't do anything more. We just let it be. We collected that HCD scan. If it's an O-glycopeptide, our search engine, our real-time library search is telling us, all right, this looks more like an O-glycopeptide. We're now going to collect an ETHCD scan on that species. The rest of these ended up being classified as N-glycopeptides, so we didn't take any ETHCD scans. So you can see now we're collecting multiple MS scans on only precursors that we think are O-glycopeptides, whereas the N-glycopeptides just stick with that collision-based fragmentation. And so, I actually neglected to show this or talk about this part in the earlier slides, but if you look at these two methods here, if you think about the number of unique glycopeptides you can identify, with the stepped HCD methods, you can get further into the glycoproteome because you're just moving faster and able to take more scans. And so when you do this collision plus electron-based fragmentation, you lose out on some identifications because you're just going slower. The benefit of this middle method here is that you get O-glycopeptides because you really can't comment on O-glycosylation when you just do collision-based fragmentation, but you're able to localize a fair number of O-glycopeptides when you include that ETHCD. So our goal with ADS would be like, well, how can we get the best of both worlds? How could we maintain that high level of identifications with the collision-based fragmentation for N-glycopeptides, and then stack this teal bar on top, and so we can get the O-glycopeptides without sacrificing the N. And so with ADS, it didn't quite, and it's not perfect, right? But it gets closer to that idea where you can maintain a relatively high number of N-glycopeptide IDs while also saving and salvaging a good chunk of those O-glycopeptide IDs by doing this real-time library search. And so this paper came out at the end of 2024, and this is actually the only published data I'm showing today. So all the other stuff is unpublished currently. But if you want to read more about this, it was a relatively straightforward story of how we could approach this and use the idea of ADS to improve N and O-glycopeptide characterization within the same complex sample. And again, if you have the Orbitrap Eclipse or Ascend, you should be able to try this on your own if you are interested. Okay, so we've talked a little bit about informatics and Glycounter. We talked about ADS and how Glycounter kind of helped inform what ions were there and how we could use those. And so I want to then now show an example of how we used ADS for some interesting sample preparation. And so we'll dive into that now and want to kind of show how useful this type of real-time library searching can be. First, I'll say, when you think about, let's go back a slide. Think about sample preparation. A huge part of the glycoproteomics workflow is what enrichment method will you use? How will you fish out glycoproteins from the mess of the proteome that exists in a complex lysate? And so, there's a whole host of these enrichment methods, and none of them are perfect, right? This is one of the challenges of glycoproteomics is that because glycans are so heterogeneous, so are the enrichment tools. And so it's hard to pick one enrichment tool that is broadly great for everything. So that's why we have this suite of tools that you can pick from and choose like, well, if I'm trying to look for a certain glycan feature or not, I can choose the best enrichment method for this. And so we were curious, as you've heard recently in the proteomics world, there's been a lot of push about nanoparticle enrichment, basically forming protein coronas on nanoparticles. And this helps enrich lower level proteins that are in plasma. And so because that protein corona forms, it doesn't always form in with the same dynamic range that exists in plasma. So you can actually dig deeper into this. So our question became like, does this selectively enrich for certain types of glycoproteins, or how could we understand what this enrichment looks like if we wanted to add it to the toolkit of different enrichment methods? And so first, I'll just introduce this workflow that I have a feeling if you've been in the proteomics space, you're probably quite familiar with. But Seer is this company that has a proteograph workflow. And so this workflow I'll describe as what proteograph is. The idea is they have surface functionalized nanoparticles, and they can incubate that with the sample of interest. And again, most often this ends up being plasma. I'll show plasma data today, although we're also exploring some cerebral spinal fluid enrichment with this too. So, we're trying to understand what it can get in a lot of different matrices. Okay, so you add your particles to your sample, you incubate, and then you use a magnet to basically just try and get rid of everything else in the plate, and then you have your protein corona from there. And then the protein corona can be digested off bead. And so you have all these different, maybe potentially nanoparticles with different surface functionalities. They can get you different types of proteins that are enriched. So you do this enrichment, and then you can go do LCMS that way. And so this exists. This is not anything our lab developed, but we were curious. We kind of talked with Seer, like, I bet there's something interesting we could understand about the glycoproteome, especially in plasma, with this. So we kind of reached out to them to talk more about what this could look like. And so in the experiments that we did, we decided to look at plasma again, same matrix that's been published on quite a bit, and to look first at a no nanoparticle enrichment. So if we just did a plasma protein digestion and ran it on LCMS. Like, what does it look like? We also then would do a glycopeptide enrichment. In this case, we're doing a strong anion exchange Ehrlich type of method, or it's really closer to a mixed mode anion exchange, so MAX. But so we're kind of doing this enrichment after we do the digestion. Again, right, and this side of the graph, we have no nanoparticles involved whatsoever. We also then had samples from Seer that they had enriched with nanoparticle A and nanoparticle B, and those are two just different mixtures of surface functionalizations that Seer has created through a lot of proteomics work that they've done. And so we'd get the nanoparticle enrichment and do the digestion on bead. And this is actually where we would get the sample from Seer. So we didn't touch anything up through the whole proteograph workflow. They just sent us, we sent sample, and then they sent digested peptide back that had been enriched with nanoparticle or had not. And then we would add this glycopeptide enrichment layer. So we would look, first of all, do we see glycoproteins from nanoparticle enrichment without any glycopeptide enrichment? And after we then enrich that nanoparticle enrichment further, do we see any benefit in the glycoproteomics workflow? So there's a kind of a lot of moving parts to this, but this hopefully a comparison of what Seer gets with nanoparticle enrichment versus what you might see in the plasma glycoproteome without nanoparticles. With this is one of the reasons I wanted to introduce ADS first, is we chose to do ADS for this because we know that there are O-glycoproteins in plasma as well as N-glycoproteins. And so we chose this method, and hopefully it can highlight the usefulness of ADS, is that we didn't have to choose to run each sample twice, especially because we had such a small amount of sample. We basically didn't have enough to inject multiple times to do, this method for N-glycopeptides and this method for O-glycopeptides. So doing ADS really allowed us to maximize the information we could get from these plasma enrichments. And just to go through this quickly, I'll show you that when you don't do nanoparticles, that's all the ones with the hatches, you can see that the number of glycopeptide identifications you get, it doesn't actually do a ton. So this would be no nanoparticle 0, and nanoparticle A and nanoparticle B. The nanoparticles themselves didn't they still had a lot of non-modified peptides in the sample. So they helped enrich glycoproteins, but because there's so many non-modified peptides, it still was hard to see the glycoproteome. When we added in this now mixed mode anion exchange, you can see those glycoproteins are definitely present in the different nanoparticle enrichments. We just had to pull them away from that really complex mixture of modified and non-modified peptides. So if we, quickly look at this, you can see like what the most unique things were and where overlaps are. I think for time, I don't have, we can't go through this in detail, but just want to point out that we did all these comparisons and also tried to look at these different nanoparticle versus glycopeptide enrichment, what types of glycans that we saw. And so you can see the nanoparticle B plus the max enrichment saw the most types of glycans, whereas, if we only had certain nanoparticles included, maybe we preferentially got some glycan classes or another. I'll mention too, just that we look at the abundance here. So this is PAX-DB and the plasma glycoproteomes. This is the rank of just the abundance there. And if we look at nanoparticle without any no nanoparticle data, nanoparticle A only or nanoparticle B only, these are glycoprotein identifications. You can see kind of where they are on this curve. And so this is a comparison then of like if we start doing nanoparticle plus glycopeptide enrichment, how much deeper and more sensitive we can get with the glycopeptide enrichment, and then also how this compares if you do nanoparticle A versus no nanoparticle or nanoparticle B versus the other conditions, like how deep into the proteome you get. And of course, there's a fair number of shared things also, that we were looking at too. So it helps improve the lower abundant protein sampling or glycoprotein sampling by doing some of this nanoparticle enrichment. The last thing I'll share for this story is that glycopeptides, we looked at O-glycopeptides. You can see actually a fair number that we could get with the different nanoparticles relative to no nanoparticle. But when we did the max enrichment, it actually hurt us in all cases. So we're exploring some other enrichments to look into how we could improve our O-glycoproteome data with this nanoparticle-based enrichment there. All right, so this is basically the end of where we are. I have just one thing I want to sneak peek for you that's not quite ready for release, but I have one slide on it. But before I do that, just kind of, you can see hopefully how all of these things cross-talk together to help us inform from our data to our methods to the type of samples and enrichment that we're going to do. So the one thing I can show is that not only are we thinking about oxonium ions, we're also really, really interested in how do we get better at evaluating identifications from search algorithms and maybe manually sequencing some spectra that our search algorithms don't get. And so we've been working on the Interactive Peptide Spectral Annotator, or IPSA. And this was a tool that came out of the Kuhn lab while I was there as a graduate student. And so we actually, Vishnu in our group is an undergraduate student, just insanely talented, totally refactored and rebuilt IPSA to allow us a lot of flexibility with glycosylation. And so we're really excited. Again, we hope to have a video demo and things like that coming within the next few weeks and release this to the public to start getting people hands on it to explore their glycoproteomics data. And of course, it wouldn't be exclusively glycoproteomics, but it's built to be able to kind of investigate all the nuances that come with glycoproteome, complications of localizing glycosites and all the fragment ions that could or could not be annotated if you get the wrong localization. So we're excited for this. And so to put it all together, we're trying to build a suite of tools that not only help us get better at chasing some of the biological problems that we're really interested in that focus on this glycocalyx, but our goal is to get things to other people too. Like I think that's a need that is constant and has been so much improved upon in the past, handful of years of getting more tools in the glycoproteomics space. But we still need more, and we're hoping that some of these can be useful for folks as we get them released that way. So I'll say that Glycounter, forgot the ER, Glycounter is going to be pre-printed and on GitHub within the week. So it's like we're finalizing that and getting it submitted. So that'll be ready to go very soon. We're actually working on some updates to how ADS does its matching. We're still kind of far away from sharing that, but excited for that. The preprint for the Seer nanoparticles is also in the finalization stages, and we're hoping to really push on this IPSA platform to get it out to people, especially just get it released on GitHub so we can have people start evaluating and using it for their studies. So with that, I just want to, end with this acknowledgement slide again and point out some of the people. So Imajay led a lot of the work on the Seer data. Imajay and Tim worked together with Jacob on the ADS story. Katie really took charge for the Glycounter work, but everyone in the group helped analyze some of those data sets. So again, you're kind of seeing work from everybody there. And then Vishnu here was kind of leading the charge for IPSA, which we're really excited to release soon.